Hi everyone!
You're listening to SciPub,
where we are talking science.
Not only for the experts, but for everyone.
Welcome to a new episode.
Hello everyone, this is Mo and Sergi, and you are listening to SciPub. In today's episode, we are happy to present our guest, Marc Talló. He's a researcher, a postdoctoral researcher in the Virology unit in the PRBB, and he's working with mRNAs and is actually the co-founder of Nocturne RNA Therapeutics. And he will talk about the topic of today, that is virology. So, with this interview, we wanted to ask you some questions that you will see. We have interviewed some people in the street and they have also questions. And we wanted to give some advice and some general information to the public. So, to start a little bit, can you explain in your own words what a virus is and what's the difference between virus and bacteria? Because we know that some people may not be that used to this concept.
Yeah, sure. First of all, like a virus, I would say it's the most simple thing that we can find in nature, or one of the most simple things, because it's like a coverture of lipids and then some nucleic acids inside. So at the end, it's like only this, a virus. So you have lipids, DNA or RNA, and that's it. So it's an organism. This we can discuss later if they are alive or not, but I have my own opinion, but it's always tricky to say. But at the end, it's like something that on its own cannot do anything, and it needs to enter to a cell, and then once it is in the cell, like all the cells start working for him. So in this sense, I would say it's something super simple, that in its own, it cannot do anything. However, a bacteria, on the other side, it's like an organism that it's really big compared to the virus, and then that it can act on its own. So it doesn't need to infect a cell to perform its cycle. So if you remember in the high school that they said an organism has to relate to other organisms, it can reproduce itself, and the nutrition, like the three main things, a bacteria will fit on this definition. However, a virus, no. So in this sense, I would say that is if we compare two completely opposite things. It's like, I don't know, if you ask me to compare an elephant and a plant. So it's like something completely different. And the message, I would say, viruses are the most simple thing that we can find in nature.
Okay, and also we know from the pandemic especially that we are using vaccines to comfort viruses. So can you define us a little bit what a vaccine is and what it's doing?
So vaccines at the end is the way that science has found to train our immune system. So you have to imagine that our body, every time that it finds like some external element, activates the immune response, and then with this, it does like, it has to react, and it takes a time to generate the immunity, to generate a way to fight the virus in this case. However, if we do a vaccine, what we are doing is training our body with a virus, for example. So you say, look, if one day you find this virus, you already activate this response. So at the end, I would say that you have to imagine a little bit like a marathon. You would not go directly to a marathon. You would go first to do some training, and then you would run the marathon. So here it's a little bit the same. You train your body to something, and then the day that you will get this infection, for example, your body will respond much faster and will counteract the attack of the virus in a faster way than if we hadn't had this vaccine. One important thing that I have to point out here is that right now, the vaccines that we are not putting the virus inside our body. We are putting parts of the virus that are sufficient to activate this immune response. So at the end, it's like putting a small part of the virus, then you generate all the response, and then your body will be ready for whenever you have it. If we trust in vaccines and we vaccinate ourselves, what we do is like we decrease the number of people that will get sick, because at the end our body will be already ready to fight this virus. And if we don't vaccinate ourselves, you have to imagine that our body will be really slow to first learn what is the virus that it has, to generate the response, and all this. Maybe it goes faster, the virus, than our immune response to answer. So I would like keep in mind that vaccines is like a training for us, and that's why I think they are really important.
So you are mentioning that a vaccine is like a training to our body, that we are training it with parts of virus or inactivated virus, but how come after this pandemic we are listening all day the RNA vaccines, and I know this is more of your topic in the research, so can you explain us a little bit what is RNA and what are these vaccines doing to us?
At the end, I think that I will go a bit before that, explaining that we all know, like, or it sounds to us what is DNA, no? And that we have the DNA. So DNA would be like, everyone is putting this example of the book of instructions of all our body. However, as you can imagine, the cells that we have in our heart doesn't behave the same as in our eyes. So what it makes specifically, like, that each cell act as it will be, is the proteins that you have on the cell, let's say. And to pass from this book of instructions to the protein that will give the function of the cell, we need an intermediate. That is the RNA. So sometimes I like to think that this process is like when you want to cook something in the kitchen. You can have a book that is the DNA, that it's full of recipes, but if I want to cook pasta, then I will go to the chapter of pasta and I will read how to do it. So I would say that a little bit is DNA would be the whole book, RNA would be the chapter specific to do the dish of pasta that you want, and then once you do this dish, following the instructions, you get the result that would be the dish of pasta. So in this sense, it happens a little bit the same. So when we say that we are generating an RNA vaccine, what we are doing is giving the instructions to a cell to generate one of the proteins of the virus. So the RNA will give some information to the cell to make one protein. So this is also interesting because we were talking that there are several types of vaccines, that before there was like inactivated virus, so people were putting a virus that was inactivated, the whole virus. Here, as I said, we are only putting one protein, one single protein. So at the end, what it will do, this RNA, is enter to our cells, and there will be a process that it's called translation, that it will give from the RNA a protein. This protein, then it will go to the surface of our cells, and it will be recognized by the immune cells. With this recognition, at the end, we will activate the immune system and generate this training I was talking before. So at the end, an RNA, it's just the instructions for a single protein. Then I will explain you that not all RNAs are as simple as this, but in the case of the vaccines, is instructions for a protein, a viral protein.
So regardless to the types of RNA, I think we know you are working with circular RNA. So we know mainly if I a high graduate student or undergraduate, I will ask mainly we have mRNA, RNA, tRNA, but circular RNA. So what is this?
So at the end I will say, like, RNA, the group is a chemical, but depending on the shape, depending on the folding of this RNA, we can have several types. mRNA is the most typical one. However, when I started my PhD, that it was long ago, but suddenly it was discovered this thing that was an RNA that instead of being linear, like the mRNA, it was circular. So at the end it was a circle, and nobody knew what was that. Mm-hmm. And for a long time people thought it was like a defect from the cell, or it was like trash, yeah. Mm-hmm. And suddenly, like it was in 2013 that it was one of these RNAs that was described that was important for the brain development. And since then, like a lot of people start studying them. And in my case, what I did was to try to see if viral infections were changing the expression of these circular RNAs in the cell. So at the end, what we were checking was if I have a cell with the virus and a cell without the virus, the expression or the number of circular RNAs is the same or is not. Mm-hmm. And then what we saw was that in the infectious, we had more circular RNAs that were helping the virus to do its cycle. Mm-hmm. So having all this in mind, we were like really interested in these molecules, because if I go back to the mRNA, as I said, you have to imagine that it's a linear thing. And as a linear thing, it has two extremes. These extremes, like there are plenty of proteins inside a cell that will degrade. It's like Pac-Man, you have to imagine, like somebody that starts eating the RNA. However, if you imagine a circular RNA, where it starts. So this was like one of the advantages of working with these kind of molecules, because as they don't have extremes, they are really stable in the cell. So then, with this thing of stability, it's when we were starting thinking like, how can we apply this knowledge to something else? And it came back another idea that it's the one that maybe we will talk later, that it's the one that we are working in my spin-off, that was to design artificial circular RNAs. That at the end it was circular RNA molecules, highly stable, and therefore it could last longer in cells. I don't know if you want that I go deeper there.
No, that's fine. But if it becomes stable in the cell, what's it do with the virus? So what's the function actually it could do the circular RNA in the virus?
So here there are like two different things. For example, the ones that are naturally occurring in the cell.
And the artificial one you are created.
These ones, the natural ones, yes, they interact with proteins that maybe the virus need, or maybe they are antiviral and they hijack these proteins and then the virus can replicate. And this is like the normal thing that happens into the cells. However, in the artificial ones, that are the ones that we design, mm-hmm, so that they are not present in the cell, what we do is like we put some sequences in this circular RNA that will bind to the virus. Mm-hmm. And binding to this, then the virus will not be functional. You have to imagine a little bit that the virus, it gets stuck and then cannot replicate.
Mm-hmm. So it's a kind of not vaccine, but kind to stop the virus from replicate, which is more or less what the vaccine gonna do. Like you stop the virus to replicate inside the cells, so you don't have more replicates. It's not gonna spread to the other cells around. So our idea, like we always thought on this like a treatment, because a lot mm-hmm, of people talks about vaccine, and at the end vaccines is something that we do in a preventive way. So even before having an infection, we prevent or we do this prophylactic. However, I mean, we've seen with vaccines, there's people who has the disease. I mean, it happened to us, like even if we were vaccinated, people got sick. So in Nocturna, what we're interested is in the treatment. So once you have the virus, how can we treat this? So at the end, with the approach that Nocturna we thought there, the idea is that we want to treat cells that they already have the virus. Mm-hmm. So in this sense, it's a new treatment. At the end, we think it's everything important, like prevention and treatment, because even the prevention works. Mm-hmm. There's always like elderly people that are in risk or immunocompromised people, so treatment is important. Yes. It's true that right now what we are trying as well is to see whether it would work in a preventive way as well. Mm-hmm. So if it could act a bit as a vaccine, mm-hmm, but this was not the goal of the So it's like a treatment, at the end of the day you stop the virus to replicate and that's good. That's fine. That's fine.
Sorry.
So you tried this or you tried this with different viruses, or it could work with all of the viruses? So it has a category viruses, or any virus could work with?
So in my case, what we are working is, if you remember at the beginning, I said that viruses are like lipids with a nucleic acid inside.
Yes.
This nucleic acid can be DNA or RNA. So the virus that I'm working with are RNA viruses. And what we did was to test with several viruses that could do a chronic infection, acute infection, because we wanted to test the potential of our technology. And as I said, if you imagine the circular RNA, what we can do is change this sequence here. So if we put sequences against hepatitis C, it will recognize only hepatitis C. If we put sequences against SARS-CoV-2, it will recognize SARS-CoV-2. So at the end, just to name a few, we started the project with hepatitis C because it's a virus that is well studied and as a model system works quite well. Then we did chikungunya, dengue, West Nile virus, that are emerging viruses, that are those ones that are transmitted by mosquitoes. And right now it was focused on some endemic areas, but now they are spread worldwide. And then the pandemic came and we said, let's try if it works with SARS-CoV-2, and we also saw with SARS-CoV-2. So
is the idea of no-cut RNA company, so you are a co-founder of this company, is coming from this applicable research, or how the idea is coming?
So at the end, I think that, I really like science. But sometimes I was missing, like, what was the connection with the society?
Yes.
No? And suddenly we had a nice project that could connect to society, could connect to help people. And then it was the moment that we said, what if we try to change, like, instead of a research project, we move to something that I hope it will reach a patient and it will help to improve the treatment on viral diseases or to the antiviral drug development.
It's like production, you have a product, or it's a link between a research and patients?
So at the end, our idea would be to generate a product that then we can bring to patients. The plan, so this will be the plan. However, we are a small startup, so what we are focused is in the R&D, so research and development. So at the end, it's like trying to improve our product to something that could be sold to a bigger company. But at the end, I don't want to go against all the researchers, because science in the world right now, it's amazing what it's doing. But it's true that sometimes I think that, and this is a criticism to scientists, me included, but sometimes we get obsessed with a protein or something, and you forget about the big picture, or how the knowledge that I'm generating can help to the
applicable.
So in this sense was a bit like my I don't know, this moved me, you know, and I wanted to translate a little bit my research to something. And I got the chance of finding during the path my PhD director and other partners that at the end we decided to found the company. And I hope it will go to a nice direction.
So talking about translating things to society, we want also in SciPub to do the other way around. So as I said, we ask questions to the society, and there are some people who have some concerns, let's say, about vaccines and mainly about the pandemic also. So we wanted to discuss some questions with you. So one question that was brought up was, why was the COVID vaccine production so much faster than the other, like the previous vaccines in the market?
Here I have to be honest with you, there was like a massive interest, like economic interest, I would say. In the sense that all the world stopped, so all the efforts were put together to move to this development. So in this sense, once you put a lot of money and a lot of people working with something and collaborating, because this, I think it's important, how the scientific community work together to try to go fast and to find this. So first of all, there was like the resources available for these people. Then it's also that it was a virus from a family that we knew before. So with the SARS-CoV-1 epidemic, there was a lot of information about the virus that we already knew, so that we didn't have to start from the beginning. It was like we started a little bit higher, and then there was all the approval of the FDA, so the Food and Drug Administration, or the European, the EMA. So the agencies, they shorten, let's say, the processes, always controlling a lot, like all the drawbacks, all the side effects, everything. But it's true that as there was an interest, they tried to do a process that was longer in a shorter term. You have to imagine that when we say that a drug needs 10 years to be approved, is because it's like one phase, then they study the results of this phase, then they planify the second phase, it starts the second phase. Here it was like, while they were doing the phase 1, it was already being discussed how to do the phase 2. So at the end it was a fast process. But as I said, it was because, I mean, we couldn't, I would say that if we had waited longer, I mean, economy would have crashed, and like people would have like, it was hard, the pandemic, for all of us. So I think that there was a need to be fast, and then like all the bureaucratic stuff was also done faster in order to help this to be one of the fastest developments, let's say. Just to mention here, for example, it's the first mRNA vaccine approved. And for a long time it was discussed if it had to be approved or not, but suddenly you have the need, and it's or this or nothing. So then sometimes we have to go to the most extreme scenario to advance in science, let's say.
Yeah, and also the technology, I guess it was not new, the mRNA vaccines.
So the like there are all those groups, like the ones that so mRNA is a field that it's been studied for long, like lots of years. So this knowledge was already known, like that. Then people working in vaccines was a field that it was a lot of infrastructure and resources. So at the end was putting the pieces together, and it's true that there were like some companies that were faster than others, but at the end it was like a race, and with the help of all the administration.
So for those people that doesn't trust vaccines or are not pro-vaccines, what would you say, or what would you?
So, I mean, here...
I know this might be a little bit different.
No. I will try to be polite, let's say. But, I mean, let's go to the data. So, we have eradicated diseases thanks to vaccines. For example, smallpox. We can see the number of cases of COVID-19 before and after the vaccines. We have seen that the countries that they don't vaccinate, the number of diseases that were not eradicated but forgot are coming back. So in this sense, vaccines work, and this is a fact. For me here, what I'm more concerned is that people is really worried of, oh, now I'm putting an mRNA. But when you were a child, you put all the vaccines and they were worse vaccines because it was like proteins or parts of the virus. Here is like, as I said, the instructions of one single protein. So in this sense, it's like, I don't understand why people is so critical with vaccines, but not with other kind of drugs that they can take. And drugs in the sense of medication, not like, but at the end, in this sense, it's like, check the data. And I think that this is one of the most important part of scientists, to communicate the data, but also to public audience to check the data. And it's seen and it's been demonstrated that vaccination programs work.
Yeah, but I think the problem with some people is not if the vaccine will work or not, but after it's work, what it's actually do in my body. So the idea is, if vaccine work, fine, but I put something, I don't know, in my body, and I don't know what this actually do after he could doing the vaccination. So they are have concern about other effects of vaccine in their bodies, not actually the main effect. Okay, this is work with the data, but if there is any other effects could happen when they are take the vaccine. So I think this is what people they are concerned.
So I understand. I mean, I understand the concern, and it's normal. But now, if I ask you, for example, last time that you take some painkiller, did you check all the side effects that you could have? I mean, like, people are not checking this, and maybe there are some of them that when you read the side effects, you would like become crazy, no? But then here, I mean, I understand it's something new, and novelty makes us to have some fear. But like, I think that it doesn't change anything. It's a chemical molecule that will be in our body. It's true that right now, like, we have a long-term study, so not really, I mean, not as in a painkiller or something like this, but it's true that we can see now which are the effects, which are the side effects, which are the recommendations for some people, and this, I mean, I'm not an expert on side effects, but it's true that I would say the WHO, the World Health Organization, has a webpage on all the recommendations and all the side effects and everything. Also, here in the building, there's ICE Global, that it's a Center of Public Health, that they did a lot of recommendations and a lot of studies on the consequences. So at the end, people are controlling this. And this, I think that it should reassure us that it was okay. And I remember that when it was the process of development of the vaccine, there was a moment that the vaccine of Oxford was stopped. And yeah, it was a pity that was stopped, but if they stopped, it's because it was not safe, or they saw something that was not as it should be. So the mechanisms of control work. So in this sense, I would say, like, trust in science, it's true, until maybe in 20 years or in 30 years, we see an effect of something. I mean, right now we know how bad is to smoke, or how bad is the exposition to pollution, but we didn't know but at the end, it's like, if we don't vaccinate, there are lots of diseases that come back, and I mean, maybe we will die even before the side effects that
the vaccine, yeah.
But I mean, as I am always saying, these organizations such as the WHO or others, they try to communicate a lot. So I would say that for these people that is a skeptic or has concerns, like, I would say read, because it's like in a plain language, not really complicated. I'm not saying that they have to read
Publications.
Yeah, but at least to inform themselves.
Listen to our podcast.
For example.
Also, like, as you super well explained, there were some people concerned about that the pharmaceuticals might have economical interest on selling the vaccines, but as you mentioned, like, there's third organization or public organizations who are controlling all these processes. So probably people doesn't have to worry that much about if the pharmaceutical company wants to sell more vaccines than are required, let's say.
Here I wanted to say that, I mean, the need of a vaccine was something that everyone was saying during that time. Like, everyone was like, we need this, we need this, we need this. The thing is, like, if a company invests its resources to generate something, it's normal that they want a profit. I'm not justifying it, because at the end, and here it's my personal opinion, but I trust in the how it's called this. So to start doing public pharmaceutical companies. So I think it's important in cases like vaccines. It would be amazing, but countries don't have the infrastructure to do it, or they don't want to do it. So at the end, thinking on the situation right now, you see this, like there's a company that produces a vaccine. We can agree on the price that they put to the vaccine. And this here, it's what my criticism sometimes. But at the end, there's like a lot of people working to get these vaccines. So at the end, there's an interest, as in all the companies. But this interest was not the one that made us to do the vaccines. No, there was a need of the vaccines. It's true that at the end, companies, and here there's like this phantom of, pharmaceutical companies are the devil. I mean, at the end, they are a company and they want to generate money. So it's true they will decide to go for something that will give a profit to them. And there are diseases that no. But this I would say goes for another direction. In the terms of vaccines, I think that, there was a need, and suddenly the pharmaceutical companies that had the infrastructure, they did it. But it was not like, we will invent this pandemic to get money.
Yeah, exactly. Understood. So from virology field, how it started, what's your background, how you started your path, how you found your interest during your career. So can you tell us like a brief about this?
Yeah, I have to say that I'm not the best example of person to... my past was a chaos, but I think it's nice to explain. So when I was in the high school, I wanted to be a journalist. And I was like, I knew crystal clear that I would be a journalist. Yeah. And suddenly, like, because of the Spanish system, I decided to prepare myself with the scientific background, because I like it, chemistry and biology. And at the end, I was like, well, I need the highest mark possible. So, but while I was doing biology and chemistry, I was like, I like that. Mm-hmm. So there was the moment that I finished my exams and the high school, and I had to decide. I was like, journalism, biology, what should I do? And one day I woke up and I said, let's put human biology. And I have to say that I was happy with my decision, but all my life I had this thought here, like, what would have happened if you had chosen the other one? But well, then I started human biology here in the UPF. And I studied, the degree, but during summers I wanted to know if I liked that, or so I started doing some summer internship in the cytogenetic lab in the Hospital del Mar. So it was a clinical lab. They were doing diagnostic, and it was not bad, but it was like really repetitive, everything. But I learned a lot, but then I realized that was not what I liked the most. And then I did my Erasmus in Germany. I went there to do my bachelor thesis, and I was also in a clinical lab. And there I realized I was doing cancer and genetics, and then I realized that my mindset wanted to understand why things happen. Mm-hmm. And the clinical data was the other way around. They wanted to categorize like the patients to find correlations, but I wanted to understand why this was happening. Yeah. And suddenly I got the opportunity to come back to Barcelona, and then I came here to do the master and a PhD. And here it was funny because all my background was in genetics, cancer, and suddenly I jumped to virology, RNA, and then it was the moment that this paper I mentioned before of circular RNAs came out, and I was like, why not? And with my director, we started discussing a lot with circular RNAs, and it was when I started like building up this project. And then, I did an internship of my PhD in Jerusalem. So I was there in a lab, and it was I really like to go abroad, and I would say that people who do science should move. Because it's a nice experience to meet new people, to see how other people work, and to work in different environments. And then I start realizing that I was loving what I was doing. And then after my PhD, I was combining this with teaching in the university. That is something that I really like. And then it was the pandemic and all this idea of creating a company. And now, let's say, I'm finishing the postdoc and I'm moving to be full-time in the company.
In one word, how was it?
So,
in one word.
Oh, it's difficult. I would say perseverance.
Mm-hmm.
Because, I can justify myself or not.
As you like.
No, but what I mean is it's a hard process. Doing research is sometimes frustrating, and then you got a good result or nice result, and then you are super excited, and it's like a roller coaster. Roller coaster would be also another word too, but at the end you start living with this. I like roller coaster. Mm-hmm. And I would say that this process was like these moments that was going down, moments that was like excitement, but at the end, I don't know, it goes like around, but you stay at the end at the same place, no? But knowing more.
I like the explanation.
Yeah, and it's very interesting all that you mentioned that you wanted to do journalism before, but then you continue to science. And also it's interesting for us because we want to encourage more like a young audience to join the career path of science. So I like that you said that was a roller coaster and it's not bad, and it was maybe not your first option in your young ages, but
at the end, one thing that I'm always telling to myself and also to the students that I have, and is life is long. So if you try a path that then, it's not the best one, you can always go back and take another path. But this other path, you will have the experience from the one before. So in this sense, I don't want to seem like, oh, Mr. Wonderful here, but it's like, sometimes I did stuff in my life that then I regret, but of course I learned. If I hadn't been in the clinical lab, I wouldn't have noticed or learned that I didn't like that. So at the end, I don't think I lost my time there. It's like I closed one door of something that I didn't like or I didn't want to proceed. So in this sense, my advice a little bit would be, don't be afraid of do it. And then if not, you go back and you try another thing. Now maybe I could say, I would have tried, or I would have done another master, but I did that one. If one day I want to do another one, I can do it. But it's not that once you get stuck to something, you have to stay to that. And I think that this sometimes is like, we have a mindset that it looks like, no, I have to do this and that, but maybe it's not a linear thing. And I think this is important for everyone.
Yeah, it is. So I just wanted to ask for some advice for the public at the end, but you already gave super excellent one. So thank you for this.
You're
talk.
welcome. Thanks to you.
Very nice to meet
interesting.
you.
Thank you. Happy to be here with your podcast.
Yeah, happy to be here too.